You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts, and I have Adrienne Scheck. She's the author of Cancer and the Ketogenic Diet. She's a PhD, senior research scientist in the Institute of Molecular Medicine at Phoenix Children's Research Institute, Phoenix Children's Hospital, and a research associate professor at the University of Arizona College of Medicine in Phoenix. She's also an adjunct professor at Arizona State University and an associate investigator in the cancer biology program at the University of Arizona. And there are many more accolades after that. So before I bungle them, I want to introduce you, Adrienne. Thanks for coming.
Thank you. It's good to be on the show.
Yeah, and just for listeners, I saw Adrienne speak at the Metabolic Health Summit, which happened about a month ago in California, and that made me more interested, even more interested in wanting to speak to her. So, Adrienne, thanks for coming. I really appreciate it.
Oh, my pleasure.
Yeah, so tell me about your work. Why the focus on cancer, and why the focus on ketogenic diets as well?
I've been a cancer researcher for a very long time. Always been interested in cancer, and I actually switched my interest to include the ketogenic diet maybe 9 years ago, something like that, because Dr. Zhang Ro, who is an international expert in the ketogenic diet for epilepsy, his lab was on the same floor as mine, and he's a clinician and a scientist, and he's excellent at both. And we started chatting in the elevator lobby, and he said, "Hey, would you like to try this in brain tumors? There's this guy in Boston doing it," and he meant Dr. Tom Seyfried, who's really sort of in many ways the father of this whole thing, I think, in my opinion. And I said, "Sure, let's give it a shot." So we kind of did a pilot experiment in collaboration. He actually had a person that was looking for a short time in the lab, so we put that person in my lab and we tried it, and the results were so good that I honestly kind of changed the direction of my lab in many ways to really start to study this. So now it's the main focus of my lab is ketones, the ketogenic diet with brain tumors.
And what the person that underwent the experiment, what kind of cancer do they have? What did they do, and what were some of the effects?
Originally, it wasn't a person. Originally, we were doing this in cell culture. So we had cells that were growing in the lab that were from the fourth tumor that an individual patient had. It was an extremely aggressive cell line. It tended to make its own growth factor, so it tended to sort of feed itself. So it was really a pretty aggressive cell line, and we tested the ketones in that cell line. So what we did was we basically just looked at how fast the cells grew by themselves, how fast the cells grew when we added ketones, and for this we added Beta-Hydroxybutyrate and acetoacetate. And then this patient had been treated with a chemotherapy called BCNU or carmustine. That was the chemo of choice back when this patient was alive. So we tried that on these cells, and then we tried the two together, ketones plus BCNU. And the reason I did that was because I knew that if we didn't find that ketones helped with the standard of care, it would really probably never get into a patient. And it turned out that this particular experiment, the results were really good. The ketones really enhanced the activity of the chemotherapy. And one of the important things to me was we did this without dropping glucose at all. So these were in cells that were grown in high glucose. So that started to suggest to me that while I believe the glucose is very important, that it's also the ketones that have their own utility. And the fact that they enhance chemotherapy is really where we started to continue the work, and we started to look at it with radiation and started to find things that way.
So what's your thoughts or your experimental evidence that how do ketones affect someone when they're undergoing chemotherapy or radiation? What are they doing to the cells, the normal cells versus the cancer cells?
That's a lot of basic research, so I don't want anybody to think that we know this in people for sure. What our kind of working hypothesis at the moment, and we're just finishing up a paper, the first paper on this, is based on animal studies in mice and based on cell culture. So it's not direct evidence in people yet. But it seems like the ketone, Beta-Hydroxybutyrate, acts as an epigenetic modifier. So epigenetics is very popular these days, and it's really a way that the cells can change the way they use their genes without necessarily changing the genes themselves. And it turns out that one of the epigenetic modifications that's important is something called the histone deacetylase or HDAC inhibitors. And people are actually looking at pharmaceutical HDAC inhibitors as a cancer therapy. And what we and others have found is that the ketone beta-hydroxybutyrate acts as an HDAC inhibitor. So our thought is that it's probably changing the expression or the use of a whole bunch of genes and potentially changing how well DNA damage is repaired. So radiation and the chemotherapy that's used in brain tumors both damage DNA. So if you can reduce the repair of that damage in the tumor cells, you're going to make those cells more sensitive to these therapies. And that's what we think is happening.
When you, do you know if there's a difference between someone, creating ketones because of their diet versus taking exogenous ketones? Has that been looked at?
Honestly, I don't. We haven't looked at that. And I know Dr. Dom D'Agostino and Dr. Angela Poff. Dr. Poff is the one that does most of the cancer work, and she's done some very nice work looking at various exogenous ketones. It's something I would like to try specifically for radiation sensitization, but there's a number of things that are different. One of the differences is that exogenous ketones raise ketone levels for a short period of time, very short, few hours, and then they come back down. So we don't know whether you need that higher level of ketones all the time to see some of these effects or not. We don't know how long the ketones have to be high. We don't even know how high they have to be. So there's a lot that is not known about exogenous ketones in terms of using them with therapy. And it is something that we plan to investigate, specifically using our mouse models as well as looking at it in cell culture. When we add it in cell culture, we know they last for a few days, and it definitely enhances radiation and chemo in cell culture. And it definitely enhances it in animals when we use the full diet. We've published that work. But whether or not giving the animal exogenous ketones enhances therapy is something that we haven't quite looked at yet.
Do you think that it causes an epigenetic change because the body would only go through that, I guess, in response to obviously environmental conditions that forced an alteration of the diet, with very little or no carbs, and therefore that's why the organism responds epigenetically?
It's a great question, and I honestly don't know. I'm kind of out of my league on that one, and I'm not sure that anybody really knows that. A lot about ketones and the ketogenic diet, I think, is really still being investigated, and it's a surprise considering how long it's been used for epilepsy. It's a little bit of a surprise, but the actual mechanisms through which it works is still not completely clear, even in epilepsy, let alone in cancer. And I think one of the reasons is because when you have these epigenetic changes, so many things in the cells change. So why the ketones act as epigenetic modifiers, I honestly don't know. And I'm not sure if anybody really has a solid answer for that one. I know I don't.
Will those epigenetic modifications appear to happen very rapidly, or does it require a period of weeks, months of continual exposure to something or a continual change in the conditions?
When we do this in cell culture, we definitely see the difference in 24 hours. We don't see as much of a change after 1 hour, We haven't done the time periods in between those two, but we do plan to. But certainly by one day of adding ketones to cells in culture, and again, this is without dropping the glucose, you see some of these changes.
Oh, I didn't catch that. So this is without dropping glucose. So the cells will be in an environment of ketones and plenty of glucose as well?
Yes. When we grow the cells in culture, normal culture media is actually very high in glucose, far higher than a person is. And when we first started doing these experiments, we purposely did not alter the glucose because we kind of wanted to see what's going on from the ketones alone, and then at some point we'll be doing some glucose experiments, just changing glucose and seeing what's happening in the cells. But we really wanted to see what were the effects of the ketones, irrespective of the effects of the glucose in the cells that were growing in the lab. So we're seeing all the changes that we've seen in the animals when we put them on a ketogenic diet, meaning the sensitization to radiation, things like that, and the slight slowing of growth of the tumor, even on their own. We're seeing those things in cells grown in culture in the absence of reduced glucose.
Oh, so yeah, what happens when you have lower glucose and ketones present around the cells? Have you done that comparison?
We haven't done it yet. I have a small lab, and we have to bring in all the money to do all the experiments. So we sort of prioritize what we want to try to figure out first. It's absolutely on the list of things that we will be doing. We will be looking at what happens when we drop glucose. But for now, we're kind of doing everything to try to get the mechanisms in regular cell culture media, which is high glucose, and then we'll basically repeat a lot of these things, adding in the reduced glucose. But for now, we're really looking at trying to drill down on the ketones by themselves.
Yeah, in an environment of ketones and high glucose, that's probably, I'm just speculating, but that's probably what would happen when you take exogenous ketones, and you either did or didn't alter your diet. You'd have that condition with ketones and higher levels of glucose. So if you figure out, if it's less than 24 hours, like you said, for the cells to achieve this protected state or for an effect to happen, maybe someone that goes to get radiation or chemo, maybe they can dose themselves with exogenous ketones multiple times, 12 hours before, let's say, and maybe that would have an effect. Again, it's all just speculation.
But that's an excellent speculation. It's exactly what we want to do. One thing I will mention is that Dr. Angela Poff has found that when you give an animal exogenous ketones, they do see a slight reduction in blood glucose, even without changing the food. And I'm not really sure why that happens, but they are seeing that there is a slight reduction in glucose just from adding exogenous ketones. But to go back to your comment, because I am now looking at all this in pediatric tumors, so I've moved to Phoenix Children's Hospital, so my work is specifically on the kids. One of the things that I've absolutely plan to test in the near future, I've actually been here less than a year, but as soon as we get some of the animal experiments going, we want to test that because it would be wonderful if we could sensitize the tumors to radiation just by having the kids drink some exogenous ketones, for example, or getting an IV of exogenous ketones for the kids where they're having trouble, getting them on a diet.
All right. So you said you're focusing on pediatric tumors now. So you said in the next year or so, what do you hope to experiment with?
I hope to see whether the things that we have found in the cells that are models for adult malignant brain tumors, if they also happen in some of these pediatric brain tumors. So we're particularly looking at brain tumors that are very deadly in children. And we've got some cells from these types of tumors, and they're very different types of tumors. So at a molecular level, they're different. So we really want to see whether this is going to possibly help inhibit the growth of those tumors or sensitize them to therapy. Because to be honest, I'm working with an awesome MD named she's actually an MD PhD, Dr. Cynthia Wetmore. And the Phoenix Children's Hospital has all the infrastructure it needs to put kids on the ketogenic diet because they're doing it for epilepsy. So it would be wonderful to extend this to the brain tumors if it turns out that ketones or the ketogenic diet sensitize these tumors to radiation also. We might be able to affect better efficacy in these kids, possibly drop the dose. Radiation is sort of a last resort for children with brain tumors because it's so damaging to the brain. So anything we can do to help reduce side effects, drop the dose, anything like that would be huge. So those are sort of our goals, is does this work in pediatric brain tumors the way we and others have shown it works in adult tumors? And then if so, can it be used to possibly get a better result in these kids?
And can you talk a little bit about the mechanism and what histone deacetylation is and does and how it affects the DNA?
I can try. It's a little hard without pictures, but I'll try. So we all think of DNA as sort of a twisted ladder. That's what the DNA molecule itself looks like. But in fact, that's not what it is. What DNA is when it's in the cell is it's wrapped around proteins called histones. So it looks almost more like a pearls on a string than it does that twisted ladder picture that we all have in our heads when we talk about DNA. And there's a lot of different modifications that can happen on this DNA. You can add different chemical modifications that change whether or not that DNA is going to be easily accessed to be read by the cell to make RNA and proteins, or if it's going to be very tightly coiled up so that it's harder to get at. And when it's really tightly coiled up and it's harder to get at, it also can be more resistant to things like radiation and chemotherapy. So by changing the acetylation on the DNA or changing the methylation on the DNA, you're changing, and these are just two little chemical groups that get stuck on the DNA, and they can also get stuck on the histones. So the acetylation is actually stuck on the histones, which is what the DNA is wrapped around. So what you're doing is you're changing whether that DNA is tightly wrapped around these little balls of histone or not. So that changes whether or not they're more or less susceptible to damage, and it changes whether genes are turned on and turned off. So without a picture, that's kind of probably the best I can do. But basically, lots of genes can get changed that way. The expression of lots of genes can get changed that way. And it's, to a large extent, that's a normal part of the way our cells regulate things. It's just in cancer, they tend to be aberrant or screwed up.
Well, what happens when a cell gets irradiated or chemotherapy attacks it? What is it? Does it, in an effort to repair itself, does the DNA, separate more to allow for repair mechanisms to happen, and then it closes back up once the repairs are done?
Well, the main thing that happens is a bunch of proteins are turned on, and they find the places in the DNA that are damaged, and they stick there, and that basically acts as a signal for all of the proteins that are involved in repairing DNA. And there's a whole bunch of them depending on the type of damage and things. But those signals basically are like standing there telling the other proteins, hey guys, come on over here, we need this DNA repaired. So it's more that the DNA can then be repaired. I don't know that it necessarily opens up or closes up specifically as much as it's turning those repair proteins on or off.
So what does histone deacetylase do? What's the mechanism by which it affects DNA?
It takes the acetyl groups off the histone, and that allows the DNA wrapped around the histones to open up a little bit. I'm not sure that I could even describe the chemical mechanism, to be honest.
Okay, but that's a fair description. Okay. So a histone deacetylase inhibitor would inhibit that from happening, and keep the DNA in an unopened state.
Yeah.
And why does this affect cancer cells differently from regular body cells?
Don't know. Cancer cells are different in so many ways. I really don't know.
Is there any clues or thoughts as to why?
Not that I'm aware of, to be honest. There might be for people who are experts in looking at the acetylation in cancer versus normal cells, but I'm honestly not familiar with it.
In what area is your specific your work specifically then would look at modulating ketone diets, ketogenic diets in order to achieve the best effect on tumors, or what's your particular focus right now?
My focus right now is as I said before, just determining whether ketones in cell culture and the ketogenic diet in animal models affect some of these pediatric tumors the same way they affect the adult tumors, and then looking at how it might be doing that with respect to what genes are being turned on or turned off, and also what other epigenetic changes are happening.
Do you think that the epigenetic changes are piling up over time? Within the first 24 hours, let's say certain things happen, and then within a week, more things happen, and a month, more things happen. Do you see an accumulation of benefit, or is it just benefit, no benefit?
We really haven't seen where it's more over long term. certainly over a couple of days, you can see a slight increase in things happening, but over long term, we usually don't do these experiments long term. Just technically, it's hard to do some of these cell experiments for weeks or months. In the animal model, we basically have kept the animals on the ketogenic diet the whole time, except for the very first experiment we did with radiation, which was published in 2012, where we gave the animals the ketogenic diet plus radiation, and after 9 of 11 animals, the tumors disappeared, and they were bioluminescent, so we could actually image them and see whether the tumor was there or not. And after about 100 days, when we were no longer seeing the tumors, we put them back on standard diet for 200 days, and the tumors didn't come back. So whatever happened in those animals for those first 100 days or so made a huge difference. Now, that was before we even knew about the epigenetic changes, so we didn't get a chance to look at the epigenetic changes and things like that. That was more sort of a phenomenological kind of thing. It's like, here's the animals, they're on the ketogenic diet. The tumor started growing, then it disappeared, then it didn't come back. So would that happen in people? Honestly, I don't know. Some of the physicians that I've spoken to when I've gone to meetings have said that when they've had patients that were just on the ketogenic diet alone, when they went off, the tumor started to grow again. There are some instances, I'm sure, that didn't happen. I don't know that people have done a real test of the ketogenic diet plus radiation where they see the tumor disappear the way we did in the mice. There's a lot of anecdotal evidence involved in the ketogenic diet, but the clinical trials are not mature enough to really answer some of these questions. You're asking great questions, but we don't necessarily have the answers yet.
Yeah, well, I'm free to speculate because I'm not doing a clinical trial. That's why I figured I'd ask you. So,
okay.
So is another proposed mechanism that perhaps the ketones are being used as fuel, and the cancer cells can't readily use ketones as fuel while normal cells can?
That was the original theory with the ketogenic diet, is that the cancer cells cannot use ketones, but normal cells can. In fact, normal cells, particularly the normal cells in your brain, sort of preferentially use ketones. They love ketones. Whether or not the tumor cells can or can't use the ketones as an energy source, the dogma is that they cannot. There are some studies that suggest maybe they can. Some cancers, not all. I think depending on who you ask, some people will tell you that they absolutely can't use the ketones as energy. Some people will say, well, the jury's still a little bit out on that. One of the things about tumors is that they are very, very heterogeneous. That's a fancy word for different genetics. So if you look at 10 people's tumors or 10 cell lines, even if it's the same diagnosis, at a genetic level, they are different. And if they're different at the genetic level, meaning what genes they have, what genes they're turning on, turning off, using, if they're different at that level, then they're also different in terms of what they are capable of physically doing. So that's called the phenotype. So, for example, there's a study that in a different type of brain tumor animal model, it's in a rat actually, that these cells are using ketones. Are they using them to make energy effectively? I'm not sure. We have people found this in mouse models? Not that I'm aware of. We're actually starting an imaging experiment with a collaborator of mine where we're going to use PET scans, positron emission tomography, where you can look at glucose utilization and things in a living animal. And we're going to look at that, and we're going to look at some biochemistry using a special type of MRI to see if we can figure out whether or not in our model, where the ketogenic diet absolutely slowed the growth of the tumor, absolutely made radiation work better, in that particular model, what's happening with the ketones in terms of, are we seeing differences in glucose utilization in the tumors? Are we seeing differences in the biochemistry? So we're doing that specifically to answer that question, is can ketones be used by these cells or not? Most people would say they can't be, but not everybody.
Are you able to mark cells and or cancer cells and then observe how they're epigenetically changing in response to different stresses, chemo, radiation, ketones, et cetera? I mean, I know we're sequencing tumors and stuff, for instance, and you said they're heterogeneous, they're not uniform, yeah. But what about epigenetically? We're looking at them that way.
Epigenetics, it's the same. Technically, it's exactly the same. The heterogeneity of these cells is also going to mean that they're heterogeneous with respect to their epigenetic markers and things. So with sequencing tumors, what happens is you take a chunk of the tissue and you grind it up and you sequence it, and you are essentially getting pretty much an average to a large extent. What some people are doing, and what we actually are just getting ready to start, is to do something pretty cool. We just got this instrument called Droplet Digital Pcr. What that lets you do is really look at kind of at an individual cell level about what's going on. And we haven't even gotten the training yet. They just finished installing it. But what you can do with that is every sample, instead of getting one answer that's an average, it actually breaks the sample up into approximately 20,000 separate samples. And there's software to do the analysis and stuff, of course. So that's actually, I think, going to get us a lot closer to the question you're asking about what's happening. Are the different cells reacting differently or the same, either at a genetic or an epigenetic level? Because we'll actually be able to take those samples and parse them out into 20,000 samples. And I think that's where we're going to actually be able to start to answer some of the questions you're asking. Because like I said, they're great questions. They're questions we've had, but technically, it's only now that we actually are going to have the instrumentation to do it. And the instrumentation to do it really hasn't been available for all that long either. So there's huge changes in technology that's going to let us start to answer some of these questions.
I guess then we could also elucidate the structure of tumors as well, because you could deliberately sample from different parts of it very finely, and you could see the evolution of the structure radially outwards or spherically outwards from the center.
People have done that. That's been published, that people have actually taken multiple samples from different regions of the tumor, and this has been done for quite a while in various different ways, where you can actually kind of see to some extent what's happening. Because the thing with tumors is that every time a tumor cell divides, it basically shuffles its genetics to some extent. It's very genetically unstable. So sometimes you can actually see which cells might have come from which cells, because you'll see certain identifiable genetic damage, and you'll see it in cells later on that also have other changes. So a lot of that's actually being done. And one of the issues, one of the problems in my mind of the people doing personalized medicine was they'll take a single sample from a tumor and then sequence it or whatever, and then say, okay, here's the therapy that's going to work, is unless you take multiple biopsies from different regions, you've only just determined what therapy is going to work on that one region, not necessarily on the whole tumor. So I think the more we get into looking at the tumor as a whole, the better all of that is going to get. And where we're going now with some of the very sensitive techniques like Droplet Digital PCR, tumors tend to shed DNA into the bloodstream. So now people are looking at what they call liquid biopsies, which is where you could take the blood and start to look at what the DNA looks like, and also how much is there of the tumor DNA. So there's a lot of questions now that are starting to be asked that way. It's not that easy to go into a person's head multiple times to get multiple samples. You either do multiple samples at the first time, or and then you can't just go back in. Obviously, you've got to, for any tumor, you kind of have to have a reason to do surgery. So this whole liquid biopsy thing, I think, is also going to really change a lot of what we think about what's going on.
Well, that's true. Do you think, I've heard tumors can come from a single cell, or they come from a single cell. Do you think that's true based on the heterogeneous nature of tumors, or do they come from multiple areas in a part of the body?
I think in general, most tumors probably arise in one place. So if you've got tumors in multiple areas of the body, that's generally metastatic tumors. And metastatic tumors are tumors where the cells have broken off from the main tumor, gotten into a transport system, for example, the blood or the lymph, gone someplace else, and been able to set up housekeeping someplace else. And it's well known that multiple cells are in, many cells are in the bloodstream of a person with cancer. Most of those can't successfully set up metastatic tumors. A small percentage can. So that's where a lot of these metastases come from. But the vast majority of the time, I think that the tumors in various parts of the body came from one original tumor. Most of the time they can find the primary. Sometimes they can't.
But even within the primary, do you think that arises from a single cell mutating, or does it arise differently?
This is just my opinion, because you can get arguments both ways, but I'm thinking it probably arises from a single cell. That when a single cell is damaged somehow to make it act like a cancer cell, and then as it divides, it'll change genetically. but I don't know that you necessarily get multiple cells close to each other being damaged at the same time. I think it's more likely that it's just a one cell, unless that person has some sort of genetic predisposition is the wrong word, some sort of genetic disease that makes them very likely to get cancer because there's some problem in their genes with respect to repair or something. And that's very, very rare. So I think the vast majority of the time, yes, my personal opinion is that tumors arise from one cell.
Yeah, but has anyone tried to calculate the mathematic, it seems like such a low probability for that to happen, for one cell to change in just the right way and then to propagate and become hundreds of thousands of cells and millions of cells. It just seems so unlikely. I mean, has anyone calculated the likelihood of such a thing?
Oh, yeah. There's been lots and lots of papers on those calculations, and the math, I have to admit, is way above my head. So you can easily do a search on that sort of thing, and you can find the mathematics involved in whether or not a tumor might have arisen from one cell or multiple cells. I'm sure that's all out there. I know there are plenty of papers that do those sorts of mathematical calculations.
Has anyone been able to find what they think is cell number one that started it all?
No. The technology is definitely not to that point.
Well, are you able to see, like you said, if a tumor metastasizes, people can find the original one, and I guess they do so by looking at the genetics of the tumors that have metastasized, and they can tell it came from, a certain one that was the original. Perhaps they could do the same within the original and find the original of the original. As you said, if it keeps mutating over time, we may be able to trace it back.
No, the way the metastatic stuff is generally done is when a tumor is found, the pathologist will look at it, and the pathologist can usually figure out what type of tumor it is. So, for example, a number of different types of tumors metastasize to the brain. So I had a neighbor, in fact, many years ago, that she all of a sudden had a seizure and went to the hospital, and they found a brain tumor. But when they look at the tissue, they say, wait a minute, this is not a brain tumor. This didn't start in the brain. This tissue looks like lung cancer. So sure enough, when they scanned her lungs, they were able to find something. But it's because the tissue actually looks different, and there are markers on the tissue, there's proteins on the tissue that they can actually stain for to say, yes, this tumor most likely came from a lung, or most likely came from a breast, or most likely came from something else. And it's really based on what the tissue looks like. Occasionally, they'll look for a specific gene that's known to be involved in a particular cancer, but more often than not, the pathologist is the one who can actually pick up on this. And now we're getting more and more information about the genetics so that there are genetic tests that can be done. But for many, many, many years, when the genetics weren't available, it was the pathologist looking at it that says, this does not look like a brain tumor. This actually looks like lung cancer, for example.
Yeah, but that's why I was wondering if you have just one tumor and it hasn't metastasized, it's the original one, and you're looking at its structure, and I guess you would think maybe the stuff in the innermost part is the original stuff. Perhaps you could see the evolution of it as the tumor grows and how it changes, and then maybe trace it back to close to its original state.
I think you can see evolution to some extent of the tumor cells, but we're not even close to technically being able to go back and say this was the original cell.
Yeah, I just wonder if we'd be able to do that or even get close. Or do you think that's still a long way off to be able to do something like that?
I think that's probably a long way off. Because remember that one original cell doesn't necessarily sit still. When it divides, it changes. So, it's not like one cell stays still and another cell is made from that one cell. Stays still meaning genetically in terms of changing. One cell becomes two cells, and the two cells that it becomes are different from the original cell very often.
Yeah, I just wonder what's the start of it. Certain mutations obviously are will be cleared up by the body. I'm sure they happen all the time, but these for some reason aren't. So I wonder how early on the immune suppression happens, or the masking of the cancer cell, and I wonder how early some of the other changes happen, and what the changes are that cause this. I guess a way to put it is tremendous success.
Honestly, I don't know exactly when it happens, and I'm willing to bet that depending on the tumor type and the genetics of that particular tumor, a lot of those things are going to happen at different times. Tumors are just it's so hard to pin down because they tend to be so different because of that heterogeneity. And I don't know that anybody has been able to trace back exactly when certain things happen in the evolution of a tumor.
Yeah, I just think it'd be really useful knowledge if we could figure that out, because it's, if you think about it, it's a bad example of it, but tumors are unbelievably successful. Cancer is unbelievably successful. It's resistant to so many things that can change. I mean, it's just, I guess that's how I characterize it, even though it's a terrible thing. So I guess there's got to be tremendous knowledge, and how does it do it,
I agree. One of the things that I kind of think of when I think about how cancers sort of do it is because they're so genetically unstable, their Darwinism speeded up. So Darwin was survival of the fittest. So in Darwin's world, of course, it takes many, many, many hundreds of years for these changes to happen. But in tumor cells, because they're dividing so quickly, if they're actually shuffling their genetics a little bit every week or every few days or however fast they're growing, that whole survival of the fittest is happening much, much, much faster. And in tumor cells, when they divide, there's a lot of cells that are essentially not successful. So a lot of cells, when they're dividing, they die. So, for example, if you get a tumor cell that splits into two cells, and one cell gets extra copies of some of the chromosomes, and the other cell is missing copies of those chromosomes, the cell that's missing some of the copies could possibly not be viable. And the cell that got extras could possibly be sort of like a Superman. It's obviously a sort of a cartoony way to think about it, but if you do it that way and you think about it that way, there's so many cells that are changing and dividing that they really have a much better chance to become so successful, for lack of a better word.
Yeah, it's just like, it seems like the ultimate form of evolution, and it also, I mean, I'm just anthropomorphizing it, but it seems like cancer is kind of like a life form. It resists death, it adapts, it wants to spread and make more of itself. It seems, it's weird. It just seems to have all those characteristics of life itself.
In essence, yeah. In a lot of ways, that's true.
All right. Well, sorry for the endless speculation, but so
that's okay.
So over this next year, what are some of the things that to recap? What are you going to be looking at over the next year, and what effects do you hope to figure out?
We are specifically looking at whether the ketogenic diet in animals and ketones in the laboratory cells that we grow sensitize to radiation, sensitize to some of the chemotherapies that are being used for these tumors. These, again, are some of the really nasty pediatric tumors. We want to look at how they're doing that, so looking at some of the epigenetic changes that happen. If ketones or the ketogenic diet can be used to sensitize some of these tumors to some newer therapies even, for example, that would be huge, because then it would be taken into the clinic. The ketogenic diet is basically a non-toxic type of therapy. And if it can be taken into the clinic to work along with the standard of care for the various pediatric tumors, I think that could be huge. So we're focusing on pediatric brain tumors to see if this is the case.
How do you perceive that the ketogenic diet or this kind of intervention is perceived by the medical industry? Are they resisting it? Are they welcoming it? Are they saying, oh, it's not a drug, so, we don't even want to think about it? I mean, what are you seeing?
All of the above. I've seen both. I've seen physicians that are starting to embrace it. So I was at the Barrow Neurological Institute for 28 years, and when I started to work on this, everybody sort of poo-pooed it. And then there was a patient that heard about it from a friend of hers, and it turns out she also knew me. She got this brain tumor diagnosis, and she came to my office and spoke to me, went on the diet, and did so well that some of the clinicians turned around and said, whoa, this is really looking good. Maybe we should look at this. And then one of the neurosurgeons there, a fabulous neurosurgeon there named Dr. Chris Smith, he's now totally on board with it. He wants all his patients doing it. He's very much on board with it, and he's a top neurosurgeon, like really, the top one there, I think, for brain tumors. So you've got some of those physicians that are starting to embrace it, but then you've also got some. I get contacted by patients all the time, and the first thing I have to do is remind them that I'm not a clinician, and then try to help them find some of the dietitians or whatever that can help them. But many times I'm told, our doctor said don't try it. Our doctor said it's awful. Our doctor said it won't help. So I think it's slowly becoming more popular. It was very, very non-accepted, very, very unaccepted. Because it's gotten more popular in the lay public with respect to weight loss and sports enhancement and things like that, it's becoming less difficult to do, which is kind of nice because there's more things available for people. And more physicians are starting to see things. The patients are saying, we're going to do it anyway. And more and more physicians are starting to see some of the benefits. So I think it's very, very slowly gaining acceptance. But there's still lots of people out there that don't believe it.
Yeah. Any anecdotal evidence of people that use exogenous ketones?
No, I have no anecdotal evidence about exogenous ketones in cancer. You might want to talk to Dr. Angela Poff for that. She's awesome. She'd be a fabulous person for you to have on your podcast. And
I spoke to her once, but I didn't know everything I should have asked her, so I'll have to have her again. Yeah.
You should definitely have her again. She's fantastic. But I think she'd probably be the best one to answer that about exogenous ketones with people with cancer.
Okay. Well, very good. Well, I appreciate you coming on. What's the best way for folks to get in contact and to find out more about what you're doing?
Probably just to look me up online. That's probably the easiest thing. So it's Adrienne C. Scheck. I use my middle initial. And a number of the talks I've given are available on YouTube so they can actually, it's a lot easier to understand it when you can see the pictures. I'm a cartoon person in my head, so that's much easier, and that's probably the best way. If anybody wants to, honestly, it's probably not efficient for them to email me and say, "What are you doing?" Because the best way to find that out is kind of online. If they want to donate to the work, Phoenix Children's Hospital, Phoenix Children's Foundation, it's a nonprofit, obviously, and they can say they want to donate to the work. Use my name. They can look up Phoenix Children's Foundation online in Phoenix, Arizona. And that'll get them to that. So that's probably the best way.
Well, that's great. Well, Adrienne, thanks so much. And I really appreciate you coming on the podcast.
Sure. My pleasure. Have a good day.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.